bodies are made out of units called cells. For example, our muscle cells make up our muscles and the skin cells make up our skin. If you cut yourself, the skin cells will destroy, and when it heals, it will grow and make more skin. Every cell in our body has a master control region called nucleus. And inside of the nucleus, there is this material called deoxyribonucleic acid. What is ribonucleic acid? Ribonucleic acid is a molecule that is present in cells of living organisms. So, every living organism has DNA that contains the genetic information and is hereditary blueprint of life. So, in this experiment, me and my fellow teammates Nabiha and Akil has prepared a DNA extraction of a banana.
List of materials: banana, alcohol, picker, zip bag, knife, cutting board, glove, spoon and filter, liquid dishwashing, salt, hot and ice water bath. First precaution: stir the salt detergent solution gently to avoid foam. Second precaution: do not keep the mixture in hot water bath for more than 15 minutes. Third precaution is do not disturb the solution after dropping the alcohol.
By adding salt, we help to neutralize the DNA charge and make the molecule less hydrophilic, meaning that it becomes less soluble in water. Then the salt also helps to remove proteins that are bound to the DNA and to keep proteins dissolved in the water. But before that can work, further explanations will be given. To break the cells to release the DNA, we need to add detergent or soap in our solution. We may have seen frequently in TV commercials that illustrate how dish soap cuts through grease, right? So, soap contains a compound that we call sodium lauryl sulfate that removes fats and proteins. Then, it will also cut through cell wall because the membrane surrounding the cells are made up of fats and proteins. The dish soap will pull apart the membranes and releasing the DNA. But before that, I will explain why we need to add alcohol in our solution. This is because the alcohol makes the water molecule less interested in interacting with DNA. So then we will see the result that the DNA will precipitate on the top of the solution. But here, why we need to add ice-cold alcohol is because ice-cold alcohol will help the DNA precipitate more quickly and also increase the yield of our DNA. DNases or restriction enzymes will destroy DNA that are present in the cell cytoplasm, and these restriction enzymes are used to protect the cell from invasions by viruses. So once we add detergents, the nuclear membrane will be destroyed by the detergents. Then the DNA is now susceptible to the DNases and quickly be degraded. So that's why we need to cool the solution, because cooling the solution will slow down the process of degradation. First of all, banana is being smashed because we want to expose a greater surface area from which we want to extract the DNA. Other than that, the physical smashing also will break the cell wall and allows the cytoplasm to leak out.